My name is Thomas Farb, CEO and co-founder of Thrive Bioscience, located in the Boston area. And we are a private company. And we started the company to automate a neglected but critically important area in life sciences, which is cell culture, stem cell culture, and experiments on cells. So cell culture is done throughout the world. It's an $18 billion per year market. It's done in over 200,000 incubators, and it's done pretty much the way it was 65 years ago, and with increasing pain points. We solve these pain points with two instruments that we'll be introducing for sale the end of this year and 2019, and they don't require FDA approval. We project revenues of over $100 million in 2022. We have significant collaborations with the Harvard Stem Cell Institute and with the Broad Institute of MIT and Harvard. Both have contractually committed to purchasing one beta instrument and 5 production instruments. We've amassed a portfolio of over 34 patent applications, of which 7 have issued, and we have limited competition. So we've raised to date $16 million from family offices such as the Estee Lauder family office, from the Milstein family office through Ogden Cap, Parker Brothers, a Monopoly game, from 7 angel groups, Life Science Angels in California, Sidecar Angels in Cambridge, and from industry executives such as the former CEOs of Novartis, SciTech, and Exact Sciences. So what is cell culture? Essentially, we're trying to create cellular environments in vivo, and we do a very poor job at it. Incubators have very little intelligence in them. They only control gases and temperature, and only while the cells are in there. We have to take the cells across multiple environments just in order to look at them under a microscope or conduct an assay. As Drug Discovery World points out, cells need carefully tuned environments, and current incubators are not capable of providing that. So our mission is to provide instruments and software tools to grow healthy and consistent cells and with data. And we're doing this with 2 instruments, a benchtop instrument and a full walk-away instrument. Cell Assist sits on the benchtop next to the incubator and complements the incubator. Currently, researchers take the cells out of the incubator, look at them under a microscope, put them back in, and almost all the data is lost. Instead, with Cell Assist, they put it into the instrument, image it, and it creates a database, it creates reports, it points out when things are out of bounds, either cells that need to be cleaned because they're spontaneously differentiating, or cases where the pH may be out of bounds. The larger instrument, Alpaca, basically has the same capabilities as the Cell Assist, uses the same optics, image processing software, sensors, optics, but wraps it in a closed, controlled environment with robotics and fluidics that act upon the instructions of the software. The Cell Assist will be available at the end of this year through an early access program with a price of $40,000, and the Alpaca will be available about a year later with a price of about 10 times that, $450,000. So why would somebody spend $50,000 to $500,000 approximately on these instruments? Well, some of it has to do with the payback, which is between 12 and 18 months. We leverage researchers by a factor of 2 to 5, depending on the processes that are being done, and we replace current existing equipment. However, what's more important is that we do things that are not currently available. So the closed, controlled environment reduces contamination, cell stress, and reduces genomic drift. The automation of protocols allows you to run the same experiment within your lab or across labs. The onboard sensors are always monitoring the growth conditions so they can be modified, as well as creating a database of all the processes that have been performed on the cells. And our image analysis reduces human error, human judgment, and it's all being done with documentation. So we have a commercially oriented team. My co-founder and chief scientific officer, Alan Blanchard, is responsible for many important inventions in the genomics field. Myself, I've started over 10 companies, such as Exact Sciences, HNC Software. And I was in the early management at SciTech, where we automated cytology labs, which is actually a really strong comparable. Cytology had been reading pap smears the same way for over 70 years. There was very little innovation in the market. And SciTech automated those labs, ultimately sold to Hologic for $6 billion, or 10 times trailing revenues. We have many of the same investors and employees from colleagues of mine from SciTech. The chairman of our board is Guy Broadbent, who ran the largest laboratory products group in the world at Thermo Fisher. We also have Brock Reeve, executive director of the Harvard Stem Cell Institute, on our board. We've now talked to over 250 customers and collaborators as part of our own market research. And there's nothing like quotes from people who are going to be using it. We have a complete lack of tools to help us in this increasingly important task of cell culture. And cell culture may be the only remaining island of outdated manual processes surrounded by an ocean of automation. We're bringing analytics, automation, and better biology to the life sciences. Thank you.
So Tom, I guess my first question is, why has there been so little innovation in adopting automated solutions in cell culture?
So it's interesting. We saw this at SciTech as well, where pap smears have been done for 70 years the same way. It's been about 65 years that cell culture has been done the same way. And I think sometimes when things have been done, that's the way we always do it, kind of a syndrome. It tends to get neglected. But I think part of the reason is also that the researchers who are having these problems are generally not engineers. They're not hardware engineers, they're not software engineers, they haven't been trained in robotics or optics or things like that. And so you have really a gulf between all the various disciplines that are needed to build an instrument like this, because we have to combine a lot of different capabilities. And I would also say that's very hard for any organization, whether it be a small private company or a large public company. It takes a lot of effort, a lot of expense to build a new analytical instrument. I also think that, again, like in the case of SciTech, most of the products that are sold into the cell culture market are sold through catalogs or over the internet. There's very few direct sales forces that ever bother to call on cell culture. And I think salespeople offer a very important feedback loop, sometimes almost like an advocate for a group, to educate the larger supplier company about the problems. And with that link missing, there isn't really that much communication. So there are many reasons why I think it's been ignored, but I think those are some of them.
So given the importance of reproducibility, how are labs addressing, improved processes to do that today that, obviously the characteristics of your product, you would help to facilitate that through automation?
So the reproducibility crisis is something that's really just being talked about. It's been kind of a dirty secret. And probably the first major piece of data came from Amgen, who did a study where they tried to reproduce 57 landmark cancer studies, and they were only able to reproduce 11% of them. Bayer, and that was in, I think, 2011, Bayer in 2012 did a similar study and was not able to reproduce 75% of them, could only reproduce 25%. And these were also landmark studies. Many others have done similar studies, including the NIH, and none have found reproducibility to be greater than 50%, which is really an astounding number. But this really wasn't talked about until recently. And I think it takes a crisis to cause us to re-examine the way that we're conducting research. And I think that crisis is really in drug development, drug discovery. The increasing expense of conducting it, the decreasing chances of success. You have better odds with blackjack than with a compound. And so many of the failures have happened in the clinical setting, and upon investigation, they were found to be the result of preclinical data that was faulty. Amazingly, the statistics are horrifying. Over 125,000 patients have been enrolled in clinical trials where the research was not reproducible. The NIH found between 10 and 20% of cell lines were mislabeled. Mislabeled. This means the researchers think they're studying pancreatic cells, but they're studying liver cells. Contamination rates have been at 10 to 20%. All of this data is really come out within the last really couple of years. And I think it's really going to be pharma and biotech companies that are licensing in that are going to drive this. For the first time, peer-reviewed journals are requiring more detailed information about how the experiments were done. So I think there's increasing pressure, but there are very few tools that are available other than writing things down in a laboratory notebook, conducting a lot of tests or assays on the work that you do, and throwing away a lot of batches. There are some companies that are increasing the reproducibility and quality of antibodies. There are a few tools around, but very, very limited, given the fact we're wasting probably $28 billion a year in preclinical research.
So you showed us your two products. You talked about the product features. As you move towards commercialization, how much of the SciTech market penetration and adoption strategy playbook do you think is applicable here?
So that's an interesting question. There are some similarities in that cell culture is a much larger market than cytology. It's also an FDA-regulated market, which is it's advantageous not to have the FDA. It also at SciTech did create a barrier to entry. But I think overall we're following, and some of this was intentional, some of this we rediscovered what we knew at SciTech. So part of the playbook at SciTech was we ended up introducing a less expensive product and then a more expensive product. And we're doing the same thing at Thrive. In fact, the first product at SciTech and at Thrive are almost the same cost, about $35,000. Our first product is creating what we think will be the first and most comprehensive, largest database of cells ever. And that actually enables many of our machine learning algorithms or image processing algorithms to do a better job. And it was the same case with SciTech. The less expensive instrument collected a lot of data, which was then used for the Imager product, which was, I think, a $250,000 to $300,000 product. I think also, just like at SciTech, when you automate a field, there's a lot of low-hanging fruit that you find just by looking at what do people do exactly, analyzing it, and seeing whether you can find a better way to do it. So, for example, in cell culture, we asked researchers, how do you know when a sample might be contaminated? What do you look for? Well, it turns out they look for what appears to them to be too rapid a change in the pH, the phenol red that's put in the cell culture media. And they kind of learn over time what to expect. They develop these heuristics for that cell line. You know, it shouldn't be becoming acidic so quickly. Well, we can deconstruct that and automate that. So our system watches the rate of change carefully in pH to see whether it's accelerating beyond what it should, adjusted for the number of cells that are growing. And if the pH is changing too quickly, becoming more acidic, chances are that there's something else growing in there. So we're doing the same thing at SciTech. We broke down each step, found a better way to do it, and found a lot of really low-hanging fruit.
And what did you understand about their current workflow that you designed into your product, such that having to deviate from what they currently do isn't a barrier to adoption?
So it's a good question, because there's very little competition, very few commercial products for cell culture. So our main competition is the way it's being done now. So it's a little bit different for each of these instruments, even though they're based on the same software. So the advantages of Cell Assist, this is a product that probably a third of the labs in the world could afford to purchase. What it allows is, for the first time, some quality, particularly for academic labs. Academic labs tend to have fellows, research fellows, international fellows, graduate students, undergraduate students, and it's very difficult for them to have any kind of standards. For the first time, they can look at who is doing what. They can review the work by each operator, each researcher. They can also supply some of the required data to journals that are now being asked for automatically. And it's, in a sense, a quality control before you run an expensive assay. Right now, there's huge variability in the cell populations, what percent are alive or dead, their health. Before running a $500 to $2,000 assay. So these labs can take a snapshot of the quality of these cells. They have a history of the cells before they run the experiment and create some standards across the lab. For the larger instrument, it really is about reproducibility and scalability. So you look at the cosmetics industry. So Estee Lauder company, like most cosmetic companies, in fact, cosmetic companies are the largest users of in vitro toxicology. And the feature that's really important that our larger product can do is operates around the clock. It runs assays periodically. So if you want to add a compound to some cells and see what the effect is, you can have time series data for, say, the first hour, every 10 minutes. You can take an image, then maybe every hour. Then you can run an assay. Then you can add more of that compound. That's almost impossible to do in cell culture, yet it's a basic fundamental thing and something the drug companies want to be able to do too in studying the effect of a compound on cells. So there's slightly different markets. Pharma, biotech, large institutes are the market for the alpaca, the more expensive instrument. And I'd say more academic labs are more interested in the features of the less expensive instrument.
Okay, well, very good. Thank you.
Thank
all
you.
for sticking
Appreciate
with
it.
us till the end here.